just to remind you what we were talking about before the break was, so we talked about different chemicals, right? And we discussed a whole bunch of them. And the last thing on the agenda to talk about is alkylating agents. For those of you who are chemical aficionados, and I know it's pretty much everybody, right? What it means alkylating? It means that the chemical that we use adds a group like this, okay? The group that consists of certain number of carbons and hydrogens, alkyl group, to the molecule of the protein or DNA. And you can imagine if such group is added to protein or DNA, it's going to change their structure. And if structure is changed, what also changes? Function, yeah. So for instance, chemicals like glutaraldehyde can alkylate nitrogen residues in the proteins or in the oligosaccharides in the cell wall or cell membrane of the bacteria, essentially linking them together. And when they are linked together, their structure changes, their function changes, they may become immovable, let's put it this way, and permeability of the membrane will be altered, or rigidity of the membrane may be altered. Okay, if the alkylation affects the DNA or a protein, DNA alkylation will cause the mutations, right? And inevitably will cause the cell death. Alkylation of proteins will alter their function, and cell will either stop growing or will die, or something bad happens to it. So what are those agents? The ones that you've heard about a lot are formaldehyde and its water solution that we call formalin. Most of the decomposition, the body decomposition, is carried out by the microbes. We have so many of them, right? We have intestinal microbes, we have skin microbes, we have microbes in the respiratory tract, and as the person dies, those microbes will contribute to the decomposition. Okay, that's why we rot. Now, we can preserve bodies from rotting either by freezing them, which is not exceptionally cost-effective way, or dumping them in the formaldehyde. So it will modify proteins, will modify a lot of body proteins, preventing them, like proteases, from acting on tissues, and it will also prevent microbes from decomposing the bodies. Okay, I know it smells funky, but nevertheless, formalin works great. The gaseous form, well, it's not a form, the actual gas, the formaldehyde, it's very toxic, can irritate respiratory passages, but it is used for sterilization of rooms. For instance, BSL-3, BSL-4 facilities that you saw fragments of a movie about. Once a year, there is a decontamination procedure, and I mentioned that. The formaldehyde is being pumped into the HVAC system of the entire suite and stays in the suite. So pretty much the rooms are filled with formaldehyde. And it is circulating through the rooms for several days. And then, of course, they kind of unplug the formaldehyde supply and they circulate normal, clean air. And then after 2 weeks, people walk in and, man, it stinks awfully. Even after, for 2 weeks, you recirculate normal air, it still stinks, and you can't really work there for more than like 2 hours because your nose, your eyes, your throat are all irritated by formaldehyde. But it's the one time if you forgot something in the BSL-3, that's one time a year you can take it out and bring it back. It's actually quite important because think about this. If you have a piece of equipment in that facility and it breaks, the repairs have to be done there in all, suited up. And you can't bring the repairman. That's the funny thing. Because in order to work in BSL-3 and BSL-4, you have to go through training. So it's going to be expensive as hell. Okay, so once a year maybe you just hold the broken piece down and then regular guy will fix it. Does that make sense? Other gases that are used for such sterilization are ethylene and propylene oxide. This is the structural formula for ethylene oxide. They are extremely reactive, okay, toxic, but very effective in treating things like medical equipment or plastic wear. The advantage of ethylene oxide compared to formaldehyde is that it decomposes much faster. So once, say, it's a plastic wear that was treated by ethylene oxide, you take it out, it's sterile, and it doesn't smell funky. Does that make sense? So pretty much all those compounds have sterilizing capacity. And it's kind of one of the concluding remarks that I want to discuss with you. Look at all the chemicals that we talked about. Things like alkylating agents or vaporized hydrogen peroxide or concentrated chlorine. Can they sterilize? Yes. Are they toxic? Oh yes. So it turns out the more toxic the chemical is, the higher sterilization capacity it has. So you can have two things together. Does that make sense? And you always have to keep it in mind. That those two properties go hand in hand. Now this table, trust me, I'm not going to ask you the main component of Lysol. I couldn't care less, to be honest. But I want to discuss with you things that you have at your house or you may buy at the store. So Lysol, let's take a look. Lysol and Clorox. Clorox, contrary to its name, doesn't have any chlorine. It has quaternary ammonia salts. Clorox and Lysol have pretty much the same main component. So they essentially aren't sterilizing. The best they can do is disinfect. And since it's a surfactant, quaternary ammonia, they will help to mechanically remove the dirt and the spoilage from the surface, right? But will this compound actually kill bacteria? It will, but not so much, because we mentioned surfactants, they aren't extremely effective. Okay? Mildew removers. These guys use sodium hypochlorite. So it's pretty much a solution of chlorine in water with sodium chloride added. That makes sense? If you ever tried to use the mildew remover on the sink, like kitchen sink, that brown slime in the kitchen sink if you don't wash it for a long time. If you spray fresh mildew remover on the slime, what happens to the slime? You don't even have to scrub it. It goes away. It's pretty strong oxidant. That's why they suggest, wear a mask, don't expose yourself, well ventilated room, and so on and so forth. So it's a strong oxidant. It's not sterilizing again. It's probably not going to kill endospores, but it's really disinfecting. Hand soaps. We talked about triclosan enough. Two things, right? What's the problem with triclosan? Huh? Resistance. right? We spilled so much of it, and the problem, it wasn't that effective in the very first place at the concentrations that they were using. So there was no point. Lysol, again, another quaternary compound. So when, when this thing, the label says kills 99.9% bacteria, it may be a little bit of a stretch. Because you remove some of those bacteria mechanically, some of them get killed, but if you have a surface of, I don't know, 1 square foot, and you have 10 million bacteria there, then by removing 99.9%, you still have 10,000 cells. So you still can spoil your food. Less chances, maybe it'll grow longer, right? Contact lens solution, that's chlorhexidine, which makes a lot of sense because your contact lens, they get in touch with your eyes. And you want to have something non-toxic but effective. Antibacterial towelettes, detergents. So again, they mostly mechanically remove microbes from the skin. Don't want to discuss triclosan again. Scope and Purell. Ethanol, again, that's not sterilizing, that's disinfecting. And ethanol is not extremely effective against non-enveloped viruses. So things like, especially with keep it in mind when, I have 2 little bottles of hand sanitizer in the car. And when we're on the road, I don't know, we do a peace stop, and we go to the restroom, and we leave the restroom, and we wash our hands, of course, but when we leave, we often use hand sanitizer. You always have to keep in mind that things like norovirus, a lot of small caliciviruses that cause intestinal distress, they're not destroyed. So wash your hands, wash your hands, wash your hands. Pine sol, phenol, and surfactant. Well, not a pure phenol, they modified forms. Okay, so it works pretty much, it has 2 modalities. One, mechanically remove the microbes, and second, destroy the membranes and inactivate proteins. I had no idea that sodium chloride is the component of the eye drops. I didn't look it up, that's my fault, but how it fights allergy is a question to me. I don't think that actually sodium chloride here is the active compound. It's probably more of a the preservative that prevents those eye drops from going sour. Okay, now this table, again, you don't have to memorize. It's a good reference, short reference, okay? I think, yeah, you will see in the study guides for methods of control that we discussed, whether they are physical or chemical methods. I want you to understand the mechanism of action. What I mean by that, if I ask you what is the mechanism of action of halogens, you tell me it's oxidation. And that should be sufficient, okay? If I ask you what is the mechanism of action for non-ionizing radiation, the UV light, you tell me DNA modification. That should be sufficient. Does that make sense? And the level of control that each method can provide. So if I ask you, which of the following methods should be used for food sterilization? And I give you options, I don't know, treating it with Clorox, dumping in the alcohol, using gamma radiation, or autoclaving the hamburger, okay? So you have to understand, since we're talking about food, autoclaving will take care of everything, including hamburger. So it's probably not going to be very, well, it's going to be digestible, just not very tasty, let's put it this way. So gamma radiation. I mean, I understand dumping it in alcohol will give it an additional modality of taste and expand the enjoyment, but again, it's not sterilizing, right? Any questions? You can expect the study guide to be up to date. Okay. So we start to talk about something that you deal with from time to time. Antibiotics. And you will notice that in this chapter, I'm going to talk about antibacterial drugs. Not antifungal, not antiprotozoal, not antiviral. Those drugs we will discuss when we will talk about relevant organisms, okay? But a lot of things, general points of considerations, will be applicable to any antimicrobial drug. And before we will actually talk about different classes of drugs, I want to walk with you through some historical points in drug development. And of course, this doesn't even cover a half of the advancements in the drug development in antimicrobials. The first antimicrobials were probably known in ancient times. Remember, we discussed those copper shavings in the wounds? That's pretty much antimicrobial. Chinese fellas were searching for antimicrobial drugs among the herbs and the plants, and you know that Chinese medicine focuses heavily on the medical properties of plants. And it's not all of it is a snake oil. Of course, the research wasn't conducted according to like rigorous medical standards and stuff like that. But a couple of years ago, Nobel Prize in Physiology and Medicine went to 3 researchers. 2 of them were awarded for development of ivermectin, the drug against helminths, and the Chinese lady was awarded for the development of artemisinin, which is not a novel anymore, but a drug against malaria. And her research was based pretty much entirely on the old books about herbal medicine, Chinese herbal medicine. So she figured out what plant was effective and managed to isolate the active compound out of that plant, which is pretty amazing. But as you can imagine, that research was not really systemic, right? They didn't have double-blind, placebo-controlled, multi-centered studies. And it's really hard to do that with antimicrobials by now, actually. We'll talk why. So first, modern scientific approach to developing an antimicrobial was applied by Paul Ehrlich, the German immunologist who was searching for the drug against syphilis. As far as I remember, it was compound number 600 that proved to be effective. That his assistant found that a compound that they later branded under the name of salvarsan was effective against the spirochete Treponema pallidum. It contained arsenic, so you can imagine that the compound was pretty toxic. But, you know, when we will discuss the syphilis and all the stages, if I would be facing a choice between the chronic syphilis and taking something with arsenic, I would take arsenic 24/7. So technically speaking, that was the first synthetic antimicrobial. But if you would look up like history of microbiology or look at the Wikipedia or look in any textbook, they will claim that the actual first synthetic antimicrobial was sulfanilamide. 3 German chemists that worked, I think it was BASF, were looking at the antimicrobial properties of various dyes, organic dyes, that company was making. And it turned out that sulfanilamide was effective against gram-positive streptococci and staphylococci. Actually, one of the first recipients, one of the first lab rats for the study of that drug was the daughter of Domag. She had a chronic wound infection, streptococcal wound infection, and she was successfully treated, and the wound actually healed completely. So that happened in, I think it was first quarter, so it took quite a while. They got Nobel Prize for that. But probably the most important discovery in the area of antibacterial drugs was a discovery by Alexander Fleming in the middle of 20s. Some books say it's 1928, 1924. It's probably taken him several years to fully formulate what he saw. Fleming returned from the vacation in the lab and looked at some old plates with staphylococci on them. And what he noticed was that in one of the plates, he saw the fungal growth and bacteria receded from fungus. So there was a clear area surrounding the fungus. I always ask students question, do you think he was the first person in the world who saw that? Huh? Say again? Oh yeah, he wasn't the first, but he was the first person to make a conclusion. He was the first to think, oh, this guy, the fungus, makes something that kills microbes around it. And that was later identified as the penicillin. Okay, so he discovered the penicillin. It took the efforts of Florian Chain, I think they were in Cambridge, to isolate penicillin as a chemical and develop the industrial method to make it in large quantities. Because the first studies of penicillin antibacterial properties were complicated. It was probably more expensive than the gold by weight. It was really like horribly expensive. So you couldn't possibly treat a sufficient number of people, but the Florian Chain developed these methods, penicillin became widely available. And three of them, Fleming, Florian, Chain, were awarded Nobel Prize in Physiology and Medicine in 1945. And I want to stop for a second. I wasn't there. I wasn't at Fleming's acceptance speech in Stockholm. But his speech became quite famous in microbiology. You know, every Nobel laureate has to deliver a speech and kind of strike some important points. So Fleming was talking about his discovery, and he also said something, warned the community about something that he already figured out is going to happen. What do you think? Huh? Yes, yes. That's what his speech was famous for. Resistance. He said, we have to be careful in using these drugs because microbes will inevitably develop the resistance. It was 1945. Everybody was so hyped up about penicillin because things that were not treatable became treatable. And it wasn't extremely toxic, you know, and microbes were responding, and the first resistance didn't show up until 50s, right? And in the 40s, pretty much 1 year after that speech, Dorothy Hodgkin, a prominent chemist, and I'm embarrassed to say I don't remember whether she was British or American. She actually established, not discovered, well, established the penicillin structure. And it was a huge breakthrough, although one might say, oh, come on, structure, so what? Bless you. By altering structure, we can alter function. We can tweak a molecule. We can change the range, right? We can change, if microbes become sensitive to one drug, we can tweak it so they will become resistant. Sorry, if they resistant to penicillin, we can modify it, make ampicillin, and they become sensitive to it. So we can constantly tweak. Okay? But you have to appreciate that it's not limitless. If the microbe is resistant to, say, amoxicillin, then you have very little treatment options. I mean, you can tweak it, but the chances are microbial cells already have enzymes that would break down this drug, that would eliminate this drug. Okay? So it was a real relief when Selman Waxman in 40s, American scientist, discovered antibacterial drugs produced by the bacteria Actinomyces and Streptomyces. Actinomycin, streptomycin, and neomycin. Neomycin and streptomycin are aminoglycoside antibiotics that are used now. They're quite effective against gram-negative microbes because penicillin is used mostly against gram-positive. You will see why. These two guys were effective against gram-negatives. Another huge another huge breakthrough, like a strategic breakthrough, was Waxman was deliberately looking for the drugs produced by bacteria. So it is a source now. Scientists all over the world look into the bacterial populations in a search of a new drug. What is the problem when you try to say you expect a huge number of soil microbes to be in a sample, right? And some of them may produce a new drug. And you want to cultivate those microbes and isolate the drug. What's the problem? You take a soil sample. You have 1,000 species there. You put it in the plate. You can streak them. If you have, new drug is like $1.5 billion. You have shitload of money. You can hire a bunch of people that will do it for you. The problem is that not all 1,000 species will grow, actually. Remember we mentioned not every microbe is cultivatable. You don't know the conditions that they require, and that's a problem. There are some developments to go around that obstacle. What actually some scientists try to do, they try to cultivate them in the soil, so they can grow in the soil. They don't change the medium. Okay? Of course, since Waxman, we had so many new developments. Drugs were modified. The mechanisms were improved. We have new drugs appearing like tetracyclines or lincosamides, or one of the latest, the drug diaryl quinolones that is quite effective against TB, because that's the main problem now, actually. Antiviral drugs against HIV. Drugs against HIV are so effective that it is now a chronic disease. So people with HIV are considered to be at the same health risk as people with diabetes or heart disease. So if they manage the disease properly, they can live in their 70s. I always say Magic Johnson is going to be playing basketball when I will be rotting in soil, So that's amazing. We made such a such huge strides in fighting microbes. There's one little problem. Their strides are longer, so we have to keep up with them. We're not ahead of the microbes. We're always following. Okay, there's some evidence that when the drug hits the market, there are already resistant strains, because it just takes time, to develop and try out and stuff. Now, general points of consideration for any antimicrobial drug. Source: natural, synthetic, or semi-synthetic. So natural drug, where does it come from? Nature. Excellent. Synthetic is synthesized. We had a discussion with the class yesterday. What is the richer source? I don't have an answer like this is the right answer, everything else is wrong. So we had pretty much the split opinion. Which source, whether nature is the better source or humans can create larger varieties. What do you think? Yeah, I bet on nature too. We were doing that for less than 100 years. Microbes were doing that for 2,500 million years. So they tried everything. They probably have it. We just can't identify it yet. Okay? Semi-synthetic is when you take the natural and modify it. Okay? Cidal versus static. Bactericidal are better, right? But if you have a healthy person, otherwise healthy person, with some kind of a bacterial infection, must you have bactericidal drug, or you can succeed with bacteriostatic? Static's fine. Well, should be okay because person has normal immunity. You kind of help immunity. When you talk about the immunocompromised patient, you want to destroy the little suckers. Now, the problem with bactericidal drugs is that they often are more toxic, have higher level of side effects. Does that make sense? Or they have other limitations. It's like you can never have all the benefits in one drug. Something is not perfect. Okay? Broad versus narrow spectrum. Each drug is good for its own purpose. When do you want to use the broad spectrum? Exactly, when you don't know what it is. When you have someone who has a headache, 106 fever, myalgia, throwing up, shitting pants, and have rash all over the body. In this case, you want to hit this person hard and hope for the cure, okay? Because it's probably something awful, absolutely awful. Narrow spectrum. You know what it is. Now, what is the disadvantage? You may say, look, why don't we just use the broad spectrum all the time? We don't even have to do the diagnosis. We just do the mass murder. That's one thing. Yes. What part? A different part. Say again? Not exactly guts per se. Huh? Your flora, yes. So you alter the bacterial composition in the guts, right? And C. diff will have a field day. Toxicity, they're all toxic on do all kinds of crap. And as you said, you stimulate the development of resistance, okay? Because, and here's the problem, if you just think about it, let's say you use broad-spectrum antibiotic, okay, which kills whatever pathogen you have, and doesn't really kill, say, some benign mutualistic bacteria in your gut. Because what they do, they gain resistance. Now, when we say resistance, when we say a trait, what determines a trait? Say, Gene, yeah. So they have a gene that actually provides them the resistance. What bacteria can do with the genes? They can transfer them. So that absolutely benign mutualistic bacteria can have a plasmid that will be acquired by, say, E. coli or something else, and will turn it resistant. So that's the problem. We always have to keep it in mind that resistance tends to spread. Okay? That make sense? Yes?
Does IV antibiotics change the microbe count like in the gut with fluid? For instance, taking orally versus taking it intravenously, are you going to change the bacteria in the gut different, or in the intestines differently?
Excellent question. The short answer, if the question is, are you going to change the bacteria in the gut? The answer is yes. Will the effect of IV antibiotics be different from effect of oral? Yes, it will be different. So antibiotics that are taken orally will have more profound effect on the gut microbiome. Now, route of administration, what Mark was looking at. So we can take drugs orally, right? We can take them IV. We can take them topically. We can take them IM, intramuscularly. So this shows you the concentration of drug in the plasma. We take them intravenously. Green is intramuscular, and blue is oral. Now, first thought when you look at it, what is the best route of administration? IV. Okay. You take them IV, but the problem is if you want to reach the gut, if there is an intestinal infection, then it may not be the optimal route. However, if you want to hit systemic infection, right? Or infection in the bones, infection in the muscles, then you want it to be IV. Does that make sense? Here, you see the concentration is higher and stays elevated for a longer time. What kind of problem can it pose to the patient? If it stays in the blood for a longer time. Toxicity, right? If it's in the blood, it may hit liver harder. Okay? Now, I had a discussion with a colleague of mine who is a clinician, Dr. Landis, and he mentioned that a lot of times the route of administration is determined not only by the disease but also by the drug itself. So if the drug cannot be absorbed in the intestines and you want to treat sore throat, then it's probably not the best choice, right? Some drugs get excreted very rapidly, so it's lower toxicity, but you have to take them more frequently. And, if you have to take like 6 injections a day, it's probably not the best idea. And we had that type of, not that type of experience, but we kind of faced it. My son had osteomyelitis, chronic osteomyelitis, and I looked at, it was very local, but I looked at the treatment options, and all treatment options said, oh, it's going to be IV for 2 months. It's pretty much wearing catheter. I was freaking out. Well, we got away with 6 months of oral. But it's, of course, from patient standpoint better, but it was 6 months. And we, morning, afternoon, and evening, we're like on the clock giving him drugs. He was fine with it, surprisingly. Another thing, you often want to combine the drugs because they may have synergistic interaction. One of the best examples is TMP-SMX, trimethoprim sulfamethoxazole. Each of these drugs is bacteriostatic by itself. When you combine them, they become bactericidal. Okay? So that's how we get the drugs out. Now, luckily, we have high throughput screening, so we can test a number of drugs for their effects in a very, very rapid fashion. And there are actually departments in the drug companies that just churn out a number of organic chemicals that they screen routinely, trying to find a new magic bullet. So this next couple of slides, they summarize basic mechanisms, basic targets for antibacterial drugs. This summarizes in the form of the picture, and next one in the form of a table. So what are the components of the bacterial cell that are present in every bacterial cell? The DNA, so we can hit DNA. Actually, DNA, RNA, all transcription stuff. What else? Not cell wall. Not cell wall. The membrane. Yes, so we can hit the membrane. Okay? So we have DNA, RNA synthesis, plasma membrane. Cell wall is in the most microbes, right? So we can try and target this. That make sense? Huh? It would be lovely to destroy the cytoplasm, but I cannot really imagine how it can happen. You can dry them out, but it's not, I mean, you can't dry out the bacteria without drying out the patient. Come on, it's written right there. Ribosomes, yeah. Ribosomes. And the great thing about ribosomes and cell wall, humans, do we have a cell wall in the cells? Cell wall, humans. No, unless you are made out of wood, you don't have a cell wall in your cells. You have a membrane, and that has important implications for these drugs, okay? We have a membrane. So cell wall, the drugs affecting cell wall should be selectively toxic. The question? Holly, Brian. Say again? So which components, which parts are present in all bacterial cells? So ribosome, DNA, cytoplasm, but we can't really target it, and the cell membrane, right? Cell wall should provide great selective toxicity. What about the ribosomes, bacterial versus human versus ADS? It's not about, they are very different. So that should give us another great, selective toxicity. We also can target some critical metabolic pathways. There are drugs that target synthesis of folic acid, which is necessary for nucleotide synthesis, and drugs. New drug that targets, it's not even on the list. The drug diaryl quinolone that targets ATP synthase. Okay. And with this, I'm going to move to the table and give you an idea. So, cell wall. Okay. 3 different types of drugs that target different steps of cell wall synthesis. You know very well penicillin. If you don't want to reveal this, it's fine. If you work in a hospital, you probably have heard about vancomycin in one or another instances. And bacitracin is a representative of the 3rd class. And we're going to talk about each mode of action separately. There's a huge number of drugs that target protein synthesis. Tetracyclines and aminoglycosides bind and interfere with the function of 30S subunit, the bacterial ribosome, and macrolides, lincosamides, chloramphenicol, and oxazolidinones, 50S. I know those names, it sounds like it's not English, but I will tell you what I expect you to know for the drugs, okay? Membrane disruption. Polymyxin B. By the way, where are you going to find polymyxin B? Can you buy it without a prescription? Or something that contains polymyxin B, can you buy it without prescription? Yep, triple antibiotic cream. So it's topical. Any idea why it's topical? Why polymyxin? Why if you will go to the doctor and ask doctor to prescribe you polymyxin B, doctor will look at you like you're absolutely nuts. What does it affect? Look at this. Membranes. Bacteria have membranes, we have membranes. So it will disrupt bacterial and it will disrupt humans. These drugs are very toxic. Has very strong side effects. Okay. Nucleic acid synthesis inhibitors. Quite effective. Can inhibit RNA synthesis or DNA synthesis. Antimetabolites, synthesis of folic acid, we're going to talk about it, and great drug called isoniazid. It inhibits the synthesis of mycolic acid, which is present in Mycobacterium tuberculosis. So it's one of the first choice drugs to treat TB. And the one that I mentioned, diryl quinoline, it's modified quinoline, which inhibits ATP synthase. As you can imagine, it's pretty toxic for humans too, because we also have ATP synthase, and it turns out they're not that much different. Okay. So first, cell wall. Before we move anywhere to the drugs, let's figure out what happens to the cell if you disrupt the biosynthesis of the cell wall. You have a cell. It's surrounded by the cell wall. The best way to assess the function of something is to break it. You don't know how your thumb is important until you cut it off. Okay? Right? So, well, look, I had the cut on the tip of my thumb, 2 cuts in a row, like bad cuts. This is so inconvenient. And it's just a cut. I didn't severed it, you know. So break something and then see what happens. So let's destroy the cell membrane, cell wall. What's going to happen to the cell? Say again? It's going to pop. It's going to pop. Yeah, it's going to lyse. Does that make sense? And when bacterial cell grows, what happens to its size? It gets bigger, right? Cell gets bigger and then it divides. And then cells get bigger and then they divide, right? When they get bigger, cell wall has to be constantly kind of synthesized. Like you're remodeling, like expanding the house. You have to build new walls, new roof, right? That makes sense? So, the drugs that contain a specific element called beta-lactam ring can inhibit the synthesis of peptidoglycan. Essentially, what they do, they bind to the enzyme called transpeptidase. Enzyme transpeptidase synthesizes the peptide linkages between the chains of the oligosaccharides, polysaccharides in the wall. Does that make sense? When they bind to that enzyme transpeptidase, which is also called penicillin binding protein, PBP, they inhibit the enzyme. Enzyme does not establish crosslinks. So you see crosslinks are missing in the bottom picture. Okay? So these chains of glycan are not linked to each other. So it's not a lattice anymore. It's just separate chains. Cell wall becomes weak. And essentially the bacterial cell lyses. Does that make sense? So these drugs are bactericidal. Okay? Now there are different kinds of those drugs. The penicillin, you'll know that. Different types of cephalosporins, monobactams, and carbapenems. And they are lined up here in kind of the order of appearance. Penicillins were discovered first. Carbapenems were discovered latest. And you see that all of those drugs have similarity in having a beta-lactam ring. Okay? What is different is this side structure, okay? And that what gives them various properties, different properties. Actually, it will change the range that they, the range of bacteria they're effective against, and resistance, so on. Even if you look at the molecule of penicillin here, you see that in the corner there's a letter R. Are you aware of any chemical element that is named R? No. Briefly, no. R stands for residue. R is used to show that something can be put in this place. Okay? And these are different somethings for different modifications of penicillin. So original penicillin, penicillin G, had this in place of R. Penicillin V, this. And then drugs that you're familiar with, I think, ampicillin or amoxicillin, are modified forms of penicillin. The latest development, methicillin, okay, was developed in response to growing resistance to these four. Does that make sense? And this, no? It does? Okay. Because another thing is, if you look at the route of administration, parenteral means not through the mouth. It can be intramuscular, IV. Probably depends on the solubility of a drug. I would strongly bet on this drug being not water soluble, so it won't be easily taken in through the intestines. Does that make sense? So we have to inject it in the blood. Does that make sense? Okay. Now, cephalosporins here, again, transpeptidase inhibitors. There are now 5 generations of cephalosporins. And each next generation was the response of humanity to growing resistance to the previous generation. But we also managed to expand the range. Monobactams, the class of molecules shown here, have produced only one drug so far, so-called S-trionam. However, carbapenems that were developed, as far as I remember, in the 90s, were thought to be an absolutely magic bullet. They are broad spectrum, effective against gram-positive, gram-negative microbes, very effective. Resistance starts. We'll chat about resistance a little bit because I want to make links to public health and economy in this case. So all those guys inhibit transpeptidase, so they destroy these links. What about vancomycin? So vancomycin prevents addition of new units of N-acetylmuramic or N-acetylmuramic acid or N-acetylglucosamine to the glycan chain itself. That makes sense? So vancomycin has nothing to do with transpeptidase. It inhibits a different enzyme, which is great, because if you have a microbe that has resistance, broad resistance to molecules containing beta-lactam, you can use vancomycin. And then bacitracin. When you build a house, when you build walls of the house, a roof of the house, you have to bring the materials in, right? You're not just bringing the entire roof, okay? Same happens with the cell wall synthesis. You have to bring in the components. And bacitracin inhibits the transport of those components. That good? So it prevents them to be transported across the membrane. So bacteria has nothing to build the wall from. We're going to talk about the resistance mechanisms. Now this summarizes the properties, and it's mind-blowing. Don't worry too much about it. So those are the penicillins, cephalosporins, aztreonam, and different carbapenems. Now if you would look at the progression of the range, you see that range increases as you go from the oldest, the penicillins, towards the newest cephalosporins and carbapenems. Does that make sense? So we focus, these drugs are effective against a wide range of microbes. Vancomycin is fairly narrow against gram-positive. Okay, it's quite effective against things like MRSA. It created other problems. And bacitracin, produced by bacillus, some species of bacillus bacteria, is also a broad range. Now, I want to discuss with you, I want you to take the stance of the microbe and think how you can become resistant to penicillin. So I have a drug, penicillin, right? Microbes, when they gain resistance to it, they start making the enzyme that is called penicillinase. What do you think it is? What does it do, penicillinase? How? Huh? Yeah, it breaks it down, yeah. It breaks it down, and there's some crap, okay? Breaks down the lactame. This beta-lactam ring, you see? It destroys beta-lactam ring, opens it up. So more broadly, those enzymes are called beta-lactamases. Okay, beta-lactamases. That make sense so far? Now I want you to step on the other side of the barricades and take a stance of a researcher. So I have a microbe that makes an enzyme that breaks down your drug. Is there any way to circumvent this resistance without altering the structure of the drug?
Break down the enzyme that the bacteria is creating.
Not the breakdown. You think, huh?
Or change it.
It's really, what can you do with how do we call the process when we stop the enzyme? What's the smart word for it? Inhibition, right? So we have to make an inhibitor for the beta-lactamase to get around it. There is an inhibitor called clavulanic acid. So often penicillins are prescribed together with clavulanic acid. So clavulanic acid inhibits penicillinase, and penicillin can work. Guess what? Microbes gain resistance to clavulanic acid. It doesn't inhibit penicillinases anymore. So it's pretty crazy. Now, I told you that I'm going to talk about the resistance to carbapenems. Those of you who work in the hospital, especially in the hospital, may have heard about the carbapenem-resistant Klebsiella pneumonia. Klebsiella is the normal component of the microbiome in the intestines, but it can cause a lot of nosocomial infections, like the pneumonia or urinary tract infections. And up to a certain point, it was successfully treated by carbapenems very effectively. But the problem now is that the new strain emerged that carries the plasmid containing the gene that confers the resistance to carbapenem. You follow me? So plasmid is bad because it can be easily transmitted between the species. And guess where this strain is coming from? The carbapenem-resistant Klebsiella. It comes from India. It actually has a name, New Delhi strain. Why do you think I stress? I love India. I love Indian food. Indians are great people, but there's some big problem there. First, too many people. Okay, so yes. So problem with the sanitation. The spread is very easy. But why resistance? The totally drug-resistant mycobacteria emerged in India. Carbapenem-resistant Klebsiella emerged in India. What's so special about India, unfortunately? Huh? They will, yes, they will. They have a general irresponsible use of antibiotics. It's expensive, and often instead of prescribing, say, 2, 3, 4 weeks, or whatever the proper course of antibiotic is, they prescribe 1 week till symptoms go away, promoting the development of the resistance. Because, and I'm not blaming. It's the intrinsic problem of a country with a population of 1 billion people. If they don't have such stringent criteria for people to practice medicine and to prescribe antibiotics. Does that make sense? So a lot of people without the proper knowledge about antibiotics start to treat folks, and oh, take it for 4 days, you should be fine. Okay? And of course, if somebody tells me take it 2 weeks and another person tells me take it 4 days, I'll take it 4 days. I'm going to feel much better. So that's the growing problem there. And Indian government tries to deal with that. Okay? Okay, I'm gonna review broadly the synthesis, and then we're going to take a break. Okay. Bacterial ribosomes versus eukaryotic are different. Okay. Drugs that inhibit the translation, inhibit the ribosome function, have very good selective toxicity. Okay. Side effects, and FYI, I put some side effects in those black boxes, okay? And I will tell you what it belongs to. Say, chloramphenicol causes aplastic anemia in the long-term run. Aminoglycosides can be toxic, nephroneuro or ototoxic. Tetracyclines. One of the most common effects is phototoxicity, increased risk for sunburns for people who take these drugs. It's long term. Okay, if it's a 1 week during Ohio winter, you're probably not having any sunburns. Now, major mechanisms. Aminoglycosides and tetracyclines bind to the 30S. Okay, so it's either inhibition of the tRNA, incoming tRNA, or inhibition of the proofreading. So no synthesis of proteins at all, or making faulty proteins. Chloramphenicol, macrolides, and lincosamides bind to the 50S. Okay, they either inhibit the transfer of polypeptide chain from one tRNA to another, so they chloramphenicol, inhibit peptidyl transferase, or they inhibit translocation, the movement of the ribosome along the mRNA, and lincosamides, I will see. And some of them, some of them inhibit the tRNA binding to the 50S subunit, binding to the site. Oxazolidinones are unique because they actually inhibit the formation of initiation complex. So they stop the translation even before it starts. Okay, we're gonna, before we go here, we're gonna take a break.